So if you see here carefully, a lot of aunties are following him, right? With this you can understand the fact that, hello everyone, this video is all about exotoxins and endotoxins. What are they and what are the differences, and all the concept behind it. So watch till the end of this video. So let's get into it. First off, we are going to understand a little bit about the bacterial cell wall. Actually, the structure is pretty much simple, right? First of all, there is a cell membrane, right, which is covering the bacteria first. Then comes the cell wall, which is made up of a thick layer of peptidoglycan, right? Now, if it's that simple, then why are we going to study about its structure and all, right? That's an obvious question, right? But the cell wall structure that we have just seen till now is one of the kind. That means only few bacteria have this cell wall structure. There are few other bacteria whose cell wall structure is a bit complex, right? I hope you're familiar with gram positive and gram negative organisms, right? If you're not familiar with it, then it's totally fine, right? It's very much easy, right? They are just two groups of organisms, bacterial organisms, which are separated or divided by using gram stain, right? Gram stain uh, divides them, these bacteria, into two: gram positive, which retain the gram stain, and gram negative, which doesn't retain the gram stain. That's pretty simple, right? Actually, the cell wall structure of these both gram positive and gram negative organisms are a bit different. So that's why they retain the gram stain differently. The cell wall structure that I've just mentioned here, this cell wall structure is of the gram positive organisms, right? It is this thick cell wall which helps in retaining the gram stain, right? That's why these are called the gram positive organisms. Now let's talk about the complex structure that other organisms, the other type of bacteria which have a complex cell wall structure, right? Till now we have seen the simpler cell wall structure which is present in gram positive organisms. Now we are seeing a complex cell wall structure which is seen in obviously the gram negative ones. Now why do these gram negative bacteria doesn't, like, why don't they retain their dye? Obviously it is their cell wall structure which doesn't allow them to retain the dye. But why, like, what is its cell wall structure? I'll explain you in a moment. Now observe this. The problem here is the peptidoglycan layer. Now I'll explain you. If you zoom it in over here, a part of it, the cell wall structure, this is the cell membrane, right? First of all, that's same as that of the gram positive organisms. First of all, there is a cell membrane. This is inner to outer view, right? This is a cell membrane first. Then look at it carefully. This is a peptidoglycan layer which is very thin, right? And this is the different one over here. This is the outer membrane which is very much peculiar to the gram negative organisms. Whereas in gram positive organisms, if you see here, there is no such thing called the outer membrane or anything like that. There is a cell membrane and there is a thick peptidoglycan layer. But here, when you see in the gram negative organisms, there is a cell membrane, there is peptidoglycan layer, but it is very thin, right? It is very thin and it is very porous, right? And there is a peculiarity over here, that is the outer membrane, which is not seen in gram positive. Now why do these gram negative organisms don't retain the dye? It is because we've seen here the gram positive organisms retain the dye because they have a thick peptidoglycan layer, right? But here in gram negative organisms, there is no such thing called a thick peptidoglycan layer, whereas here it is thin and porous, which don't let these gram negative organisms to retain the dye. That's why they are gram negative, they don't retain the dye. And here there is another peculiarity called the outer membrane structure, which is very much important to understand. So let's understand about the outer membrane structure. Now when you see the outer cell membrane, it's a bit different. Obviously it is the general phospholipid bilayer only, but the outer lip of this outer cell membrane is a bit different. If you see it closely, it is made up of a very important structure, right? It is made up of a very important structure called lipopolysaccharide, LPS. Now it is very important to understand about these lipopolysaccharide, because the lipopolysaccharide here is our real hero, that is the endotoxin, right? This lipopolysaccharide, that is the LPS, is our endotoxin. Now let's understand. We know that it's really important to understand the structure of LPS. So let's try to understand its structure. Now if you see LPS, I've drawn a cartoon type of an image like of the LPS to help you in remembering it well. If you see here, it has three components. LPS, this is a diagram of LPS. It has three components: 1, 2 and 3. From inner to outer, the innermost thing is the lipid A. Now lipid A is really important, I'll get to that again. But the second component is the core polysaccharide, and the outer one is O antigen or orthopolysaccharide, right? Now here I said that lipid A is really important to understand. Now let's see lipid A. Lipid A, it's made up of two saccharide units, obviously that's disaccharide, and it has fatty acid chains over here, right? That's why it is lipid A. Now why it is important? Lipid A is important because it is the main reason behind the toxigenicity of this endotoxin, right? Endotoxin, when it enters into the bloodstream, all the manifestations which we see are because of this lipid A. Till now we have understood about the structure of the endotoxin, that is a lipopolysaccharide. Now let's try to understand a bit about how the endotoxin itself is different from another guy, that is the exotoxin, right? Now the first thing is that we know that endotoxin is an integral component of the cell wall of the gram negative bacteria, right? Now when the bacteria enters into the human body, right, it enters into the bloodstream. For the endotoxin, as endotoxin is an integral component of the cell wall, so for the endotoxin to be released into the bloodstream from the bacteria, the bacterial cell wall needs to be disintegrated. Listen carefully. The endotoxin is inside the cell wall, right? For it to come out into the bloodstream, the bacterial cell wall should be disintegrated. Then only the bacterial cell wall should undergo cell lysis. Then only the endotoxin will be visible in the blood, right? Then further it causes all the manifestations, right? Unlike the exotoxins, right? They are just present in the cytoplasm, right? There is no need of any disintegration of the cell wall or anything like that, right? They are present in the cytoplasm and they are just released into the blood like that. They are secreted. The better word to use over here is the secreted into the blood. And there is another difference over here. When you look at it carefully, I said that the endotoxin is an integral part of the bacterial cell wall, or more precisely to say, the outer membrane of the bacterial cell wall. But when you look at the gram positive, the cell wall of the gram positive bacteria, there is no such thing called the outer membrane, right? There is only inner cell membrane and a thick peptidoglycan layer. There is no outer membrane, so there is no lipopolysaccharide, so there is no endotoxin. So from this we can say that endotoxins are only present in gram negative bacteria but not in gram positive bacteria, right? They are only seen in gram negative bacteria. You can't find endotoxins in gram positive bacteria because they don't have any thing called as outer membrane or lipopolysaccharide, right? This is a second difference. Another difference is that this endotoxin is pathogenic but not immunogenic. What do I mean by that? When gram negative bacteria enters into the human body, it when it causes cell lysis and endotoxin is released into the blood, it does not stimulate the immunity of the host. So no antibodies are formed against this endotoxin, right? But unlike endotoxin, this exotoxin, that is both, when it is released into the bloodstream, it causes both pathogenesis and it also stimulates the immunity, right? So definitely antibodies are formed against this exotoxins, released exotoxins, right? So that's why these exotoxins are they could be used as vaccines named toxoid vaccines. For example, tetanus toxoid, right? Tetanus toxin is actually an exotoxin, so that may be attenuated and used as a toxoid vaccine to stimulate the host immunity to secrete more and more antibodies. Another point is that the endotoxin, which you usually see in gram-negative bacteria, is all the same in all gram-negative bacteria. Uh, in all different types of gram-negative bacteria itself, the endotoxin is the same that is present in the outer membrane of the gram-negative bacterial cell wall, right? But when it comes to the exotoxins, in exotoxins, it is usually not the case. There are different types of exotoxins. For example, there is diphtheria toxin released by the Corynebacterium diphtheria. It is an exotoxin, and there is also tetanus toxin released by Clostridium tetani. So there are different types of exotoxins released, but when you come to endotoxin, it is the same old LPS, that is the lipopolysaccharide, which is endotoxin present in all gram-negative bacteria. Okay, now there is another thing here that is about the endotoxins, is that when endotoxin enters into the bloodstream, the symptoms that it causes is very much generalized. It is not tissue specific, right? It is not specific to any kind of tissue. It causes, the symptoms of it would be like fever and shock and hypercoagulation, something of that sort, generalized symptoms, right? But when it comes to exotoxins, it is very much tissue specific. What do I mean by that? Like, take for example tetanus toxin. Tetanus toxin, it is an exotoxin which affects CNS, right? That is tissue specific. And if you take an example of diphtheria toxin, it is also an exotoxin specific to respiratory system, right? Likewise, exotoxins are tissue specific, whereas endotoxins are not. Now the other difference is that exotoxin is a protein, whereas we already know that the endotoxin, that is lipopolysaccharide, definitely is a lipopolysaccharide, right? So exotoxin is a protein. We also know that proteins usually get denatured at high temperatures, right? So from this we can understand that these exotoxins, they are proteins, so they are heat labile or heat sensitive, right? They get denatured at higher temperature, whereas endotoxins, as they are lipopolysaccharides, they are heat stable. They don't get affected by high temperatures, right? And the thing is that exotoxin, we usually call it as potent. What do I mean by potent? Potent means small amount will also cause big damage, right? Remember this statement: small amount, big damage. Small micrograms of exotoxin is just enough to cause many uh, tremendous disease manifestations, whereas endotoxin is not that much potent. Large amounts, there should be large amounts of endotoxin in the blood in order to cause disease manifestations, right? So okay then, till now we've seen all this, like about the basic structure of the bacterial cell wall, then the differences between the gram positive and gram negative bacterial cell wall. Then we took a close look about the endotoxin structure and how it is different from the exotoxin and all those differences, right? The thing is that there are a lot of differences here, right? You may understand it properly, but the problem is that it is difficult to remember them when they are a lot, right? So I have just made a fun story to just help you remember all these differences easily, right?